So aerobic metabolism, starting off with glycolysis in this video. Now, I say aerobic metabolism, which metabolism is just the process of processing sugars, and it's also called cellular respiration. So I know the word respiration might throw you off a bit because respiration, you think of breathing with your lungs instead of the cellular breakdown of sugars, but they are related. Then cellular respiration is the same thing as metabolism, and metabolism is, of course, how your body is processing sugars and turning it over into energy for your system. Now, of course, this is aerobic metabolism as opposed to anaerobic metabolism, because as you see under metabolism, we humans, we breathe oxygen, and so we're most likely going to be in a form of aerobic metabolism. But when there is not enough oxygen available or in anaerobic organisms, after glycolysis, which we're going to study in this video, after glycolysis, you would go into the stages of fermentation in the absence of oxygen. And that's how you could process sugars. And we humans do it too. When, like, for instance, when you're out of breath, when you're exercising and you're super out of breath, you're not getting enough oxygen to your system. And after a while, your body will shift gears into anaerobic metabolism instead of aerobic metabolism. Um, so that's why they tell you to keep your heart rate within a certain guideline. And what we're going to study today is the glycolysis, which is the first step for either one, either anaerobic or aerobic metabolism. And as you saw before, for aerobic metabolism, it starts off with glycolysis, which is what we're going to be talking about in this video. And then after glycolysis, it goes into a transition stage. And then after the transition stage, we go into what is the Krebs cycle or the citric acid cycle. And then after the Krebs cycle, we do electron chain transport, and that is what produces oxygen and the most energy in the form of ATP. Now, for metabolism, as I said, it's also called cellular respiration. And there are three major steps. Some teachers in some classes say 3 steps, some say 4, because sometimes they don't count the transition step as the actual full step. So sometimes it's like glycolysis would be step 1, and then transition is just transition, and then the Krebs is step 2, and then the electron chain transport is step 3. That's how a lot of teachers and a lot of textbooks do it. So I just put a little 2A here for transition. But other teachers go ahead and say, you can say 4 steps to aerobic metabolism, glycolysis, transition, Krebs, and electron chain transport. It just depends on how your teacher is teaching it and what your textbook you're using is. The important point is that you understand that these are all separate processes and not get them confused. Glycolysis, which is what we're going to be doing today, as I said, it's for both aerobic and anaerobic processes. This is the aerobic processes, the one transition 2, 3 with the Krebs in it. So you just remember the Krebs, when they're talking about the Krebs cycle, you're talking about aerobic metabolism, cellular respiration, not fermentation. And the Krebs cycle is also called the citric acid cycle, or TCA. And so if you see TCA, citric acid cycle, Krebs cycle, that's all going to be the same thing. There's just like several different names for it, but they're all referring to that step, the Krebs cycle step. And then the electron transport chain can also be called oxidative phosphorylation. So that is the same thing also in that step. But today, as I said, we're going to be talking about the very first step, glycolysis. And as a review, glycolysis takes place outside of the cell. It goes through the cell membrane and into the cytoplasm of the cell. So it takes place within the cell, but outside any of the cell's organelles. Now, the whole point to glycolysis is that glucose is water soluble. Okay, and it's a 6 carbon chain molecule. And I drew this out for you just so you can kind of see it's just a carbon bonded to a carbon. And there's other things in the molecule as well. We're just not drawing those because they're not really important right now. I'm just drawing this is kind of like the backbone carbon chain of the molecule. And there are 6 carbons chained together. And then off of those carbons could be like other things like hydrogens and oxygens and stuff, because that's what you need to pay attention to when following along in these processes for metabolism. So since it's 6 carbons, that's kind of a big molecule and it's water soluble. It can't get into the cell through the membrane by itself. It has to have help. Well, the help arrives with GLUTs, G-L-U-T, and that stands for glucose transporters. And they do exactly that. They transport the glucose through the cell membrane and into the cell. And that's how the glucose gets into the cell in the first place. And then the rest of the steps are inside the cell in the cell's cytoplasm. Now, you should pay attention to the fact that the enzymes, I put drive reactions, but that's not necessary. If you know more about enzymes, that's kind of a misnomer. But just for beginning biology, microbiology 101, you can kind of just look at it like that, the enzymes driving the reactions. And then what is transporting the energy which is how you end up creating energy out of this glucose, are energy transporters, ATP and NADH. And for every one molecule of glucose, by the end of glycolysis, you're going to get 2 molecules of pyruvates. So for every one molecule of glucose, let me say that again, for every one molecule of glucose going in, you're going to get 2 molecules of pyruvate coming out. So that's important to remember because you're going to need to, for your tests and assignments in class, you're going to need to remember what goes in, how much of something goes in, and how much of something comes out of each cycle. So like for instance, in glycolysis, we can say like 2 ATPs come in and 4 ATPs come out, giving you a net result of 2 ATPs coming out. And you'll see what I'm talking about when we go through the stages. And I'm going to have this nice little diagram for you so you can see exactly what's going on. Okay, so I sort of wrote these columns backwards. I started over here on the right, and then I ran out of space, so I had to put the other column over here on the left. So I'm sorry if that throws anybody. But here is your glucose molecule outside of the cell. Okay, and here's your 6 carbon chain. 1, 2, 3, 4, 5, 6 carbons. And as I said, they can't get into the cell membrane. They can't just pass through naturally. So they have to use GLUTs, the glucose transporter, and it's kind of like a gate. It opens its gates and lets the glucose in. It's kind of like a pump. It pumps the glucose in. So the glucose comes in, and the first thing that needs to happen, because otherwise the glucose is just going to spill back out through the gate again because its tendency is to want to go outside of the cell. So in order to keep the glucose inside the cell and keep it from spilling back out of the gate from the glucose transporter, we need to tack on a phosphate group to that molecule so it kind of hooks it inside of the cell. It makes it stick. So here's the phosphate group over here in the black, and that's PO4 3 negative is what is phosphate. And don't worry about this too much. Just know that if you see this, we're talking about phosphate. For biology classes, you don't usually need to memorize a lot of the chemistry of things going on, so you don't really need to know why phosphate is PO4 3 minus, but if you see it written that way, just know that that's the phosphate we're talking about. That's what phosphate is. So that is step number 1. You add a phosphate group to lock it inside the cell so it doesn't go spilling back out through the GLUT anymore and go back out to outside of the cell. So this phosphate group tacked onto it hooks it inside. And then step number 2 and this is going to look exactly the same as step number 1 if you notice. Look at step number 1 and look at step number 2. These look exactly the same. That's because step number 2 is just a minor step of some other elements that are kind of branched off of the carbons that aren't drawn here. They rearrange themselves into an isomer. An isomer is it's the same molecule, but a couple of things are just rearranged. And so it just changes its name here. So it just rearranges some of the oxygens and hydrogens that are branching off of these carbons and changes the name. And that's step number 2. It's kind of a minor step. Now that we've rearranged some stuff and it's changed its name, step number 3 is to add a phosphate group to the other end of it. So now we have bookends of a phosphate group on either end of this carbon chain, and that will make those previous changes stick. So it doesn't go back sliding back into the original glucose form. Now, step number 4 is that these divide into half. So now we have 3 carbons and 1 phosphate, 3 carbons and a phosphate group. And then we have a mirror image, 3 carbons and a phosphate group. So one of these, the left side, 3 carbons and a phosphate group molecule, then changes into this right side, 3 carbons and a phosphate group molecule. It just changes back into the mirror image of itself. So we're working with all the same kind of molecule, the 3 carbons with the phosphate group on the right here, instead of mirror images of the molecule, which is what we have when we split them in half. And now step number 5 is you add a phosphate group to that to make the changes stick. So pretty much like every time you do something, you change something to the molecule, the next step is that you're going to add a phosphate group. So that kind of, if you look at it, like every time you want to make your changes stick, it's like hitting the save button when you're in a document on your computer. Adding a phosphate group makes it stick. It's hitting the save button. So it doesn't backslide into the previous confirmation. Now, step number 6 is you're going to lose one of the phosphate groups. Step number 7 is that the remaining phosphate group that was over here on the left changes over into the middle carbon. So now we have a phosphate group here, and then it rearranges a little bit and changes its name. So now we have the phosphate group still off of the middle, but the other elements have rearranged a little bit, and it changes its name. And then step number 9, put this over here. Step number 9 is that you remove that very last phosphate group, and so you're just left with that 3 carbon chain. So see, we did have that phosphate group on here. So we remove that, leaving us just with this 3 carbon chain again. And this 3 carbon chain in step number 9 is pyruvate. Pyruvate, pyruvate. But I want to go back and point something out to you. Remember when we divided this in half back in step 3? When we divide this in half, now we have 2 different groups. So everything from then on, after it's divided in half, counts as 2 molecules. So that's why we end up with two pyruvates out of one glucose molecule that we started out with at the very beginning. Now, in order to understand these processes, you need to understand some things about these energy carriers, ATP and ADP, and there's NAD plus, and the counter to that is NADH. And this carries energy in the bonds that they pick up and drop off. ATP is called adenosine triphosphate. Triphosphate. So that means it has three phosphates. So it has adenosine and then one, two, three phosphate groups attached to adenosine, the adenosine molecule. Now, ADP is the same thing, adenosine, but it's diphosphate, which just means it's adenosine, and only one, two phosphates attached to the adenosine. So it's just ATP and ADP are pretty much essentially the same thing. It's just that ATP has three phosphates and ADP only has two phosphates. So they are the same thing. They just change back and forth from one another, depending on if they pick up an extra phosphate, giving them three phosphates, so it's ATP, or if it drops off that third phosphate, turning it into ADP. So it's the same thing. They just change back and forth into each other, depending on how many phosphates they have at any given time. And if you look down here, down below this line. Here's how the diagrams are drawn. You can see this three-carbon chain right here on the left, and this is the order of the steps of what we have. So we start at the top and we're going down. We're following this arrow right here. And then ATP comes in, swoops in, and drops off a phosphate into this carbon chain. And so out comes ADP because now it only has two phosphates instead of three from the ATP. And where did that third phosphate go that the ATP dropped off? It went right here, attached itself to that three-carbon chain. So now your result is this three-carbon chain with a phosphate group attached. So if you look at it like, when your mom dropped you off at school and picked you back up from school, or maybe you're the mom or dad dropping your kid off at school and picking him up from school. Say we have this family right here, down here at the bottom, and we have the mom or the dad, which would be the adenosine, and then they had three children and two little toddler kids, and then one school-age kid who is in the front seat up here, and then the two little toddler kids are, of course, in back in their car seats. And so, say the mom takes the school-age kid to school. So you have this car full. You have A for the mom, and then you have the P that's going to go to school, and then you have the two P's that are going to stay with mom. They're going to stay with the A. And so you go and you drop off your kid at school, and then you're driving away. As you're driving away, now you have a car of just the A, the mom or the dad, and then the two toddlers that stayed with the parent. So now you only have two P's. You have the two kids in the back, but you dropped your school-age kid off at school. Okay, so look at this. So we have at the top here, we have an A and the school-age kid in the car and the two toddler kids in the car. So we have an A plus three P's in the car, right? And then you drop your school-age kid off at school. So now as you're driving away, you have an A and two P's, just the toddlers in the back because you dropped your school-age kid off at school. So now you only have two kids in the car, right? So think of the P's, the phosphates are the kids, and the A is the parent. And so you're going to either have the three kids in the car, the three P's, or you're going to have two P's in the car, two kids in the car because you dropped one off at school, right? So this top one would be ATP because adenosine triphosphate, tri meaning three, and this bottom one here, driving away from school after you dropped off the kid, would be adenosine diphosphate because di means two. So adenosine triphosphate means three phosphates. Adenosine diphosphate means two phosphates. Now there's something similar with NAD plus positive and NADH, except NAD is not dealing in phosphates, it's dealing in negative hydrogens, or hydrates, they call it. So we have this NAD plus, or positive, we have this NAD positive, it's called nicotinamide adenine dinucleotide. And for the purposes of knowing the Krebs cycle, you don't really need to know what that means yet. You will need to know what it means in further chemistry classes though, but for biology Krebs cycle, you're good, just knowing NAD plus. So NAD plus goes and picks up negative H. And so as it's driving away, as it's going away, as they picked up their kid from school, they turn into NADH. So see, when we were talking about ATP and dropping your kid off at school, you go from ATP, drop the kid off, now you're ADP. But, then it's the end of the day and you have to go back to school, ADP, you can go back to school and pick up your school-age kid, and so now you turn back into ATP. So see, you're the same family. It's the same thing. It's still the same molecule, but you change the name and you change the number of phosphates in it. And you're either dropping a phosphate off or you're picking a phosphate up. Well, same thing with NAD. you're either NADH dropping off this hydrate, this negative hydrogen, and that turns you into NAD positive or NAD plus, or you're NAD plus and you go and pick up a negative hydrogen and that turns you into NADH. And those arrows, those curvy arrows, are how it's kind of drawn on all the diagrams. So I wanted to keep that consistent so you knew how to look at the diagrams in your book. Okay, so here's the overview of glycolysis. And so, as we said before, glucose enters into the cell through the glucose transport system, or the GLUTs, GLUTSs, GLUTs, the glucose transporters. And once it's inside the cell, as I was telling you about with giving you the example of dropping a kid off at school, and you start out with 3 kids in your car, and now you have 2 kids in your car. So this goes from adenosine triphosphate, tri, that's 3, to di, that's 2. So ATP to ADP, because once it gets inside the cell, we have to add a phosphate group onto this glucose molecule in order to keep it inside the cell. Otherwise, it's just going to slide back out through the glucose transporters. So the ATP comes in and drops off a phosphate, one of its phosphates, and the phosphate joins, and it ends up, we add it right here. So now this has a phosphate group attached to it, so it can't slide back out of the cell. And the ATP turns into ADP because now it only has 2 phosphates. Then the molecule rearranges a little bit, and it still has that phosphate group on it, but the molecule rearranges a little bit and changes its name. And then, since it rearranged a little bit, we want it to stick. We don't want it to slide back into its original conformation. So another ATP comes in and donates another phosphate group, which attaches to the other side of the carbon chain. So now it has 2 phosphates bookending the carbon chain, one on either end of the carbon chain. Then the carbon chain with the phosphate group on both ends splits into 2. So it just splits down the middle into 2. So we end up with mirror images of each other, a carbon chain with, if we call this carbon 1, carbon 2, carbon 3, this phosphate group is going to be attached to carbon 1 on this one. But over here on the right, carbon 1, carbon 2, carbon 3, the phosphate group is going to be attached to the third carbon on this one. So see, they're mirror images of each other. And so this one over here on the left, where the phosphate group is attached to the first carbon, needs to take an extra step and turn into its mirror image. So we have all the same, exact same structure molecule right here. And so now we have 2 of each, 2 of these molecules, the carbon, carbon, carbon phosphate. We have 2 of them times 2. So for the rest of the glycolysis, this is going to apply to 2 molecules. Now, after we have these 2 molecules of the phosphate group attached to the third carbon, then a different energy transporter, NAD+, is going to come in and drop off an electron, a hydrate, which is a hydrate is just a hydrogen with an extra electron, so it's a negative hydrogen. And in order to make that stick, another inorganic phosphate group is just going to come in just from kind of the environment, not from the ATP, but just from the environment. Another phosphate's going to come in and it's going to attach itself on the other side, on the other end. So now, once again, we have 2 phosphate groups, one on each end of the carbon chain, bookending that carbon chain. But this time, the carbon chain is only 3 carbons long, not 6. And then after that, now we're going to start taking phosphates off. So now ADP comes in, because remember ADP, di, means 2. So only 2 phosphates come in and to pick up their kid from school. So now they have 3 kids in the car again. So that makes it ATP, because tri means 3. And so it removed that very leftmost phosphate group attached to the carbon chain. So now we just have this 3 carbon chain with 1 phosphate group attached to it, and it rearranges the molecule. Everything stays the same, but it just rearranges itself. So now that phosphate group moves over and attaches itself on the central carbon, the center carbon, instead of the end carbon, and it changes its name. And then it rearranges itself again, but everything kind of, as far as we're drawing it, everything kind of stays the same. But it rearranges the other elements, and the molecule rearranges again. It changes its name. And then another ADP comes in to pick up their kid from school, picks up that last phosphate group, and so now all we have is the 3 carbon chain with no phosphates on it anymore because we took all the phosphates off. And that molecule is called pyruvate. So let's take a closer look at these steps and get into detail about it a little bit. And then we'll come back to this chart again so you can look at it again and it'll make a lot more sense to you after we go into detail about each of the steps. So step 1 in glycolysis is first the carbon comes in through the glucose transporters in the cell membrane into the cell and it goes into the cytoplasm. It doesn't go into any of the organelles yet. It just is floating around in that gelatinous goo that all the organelles are sitting in. So it's outside of the organelles but inside of the cell itself. And then you add on a phosphate to keep it trapped inside the cell so it won't slip back out again through the GLUT transporters, and the enzyme that does this is called hexokinase. So it leaves us with the 6-carbon chain, just like glucose, but with a phosphate group attached at carbon number 6 at the end. Because see, if you number these carbons, 1, 2, 3, 4, 5, 6, the phosphate group is attached to carbon number 6. So that gives us a new name for this molecule, and this is called glucose 6-phosphate. The 6 is because the phosphate is attached to carbon number 6, just like I showed you on the previous page. 1, 2, 3, 4, 5, 6. So it starts out as a glucose molecule, but then it has a phosphate attached to number 6. So it's called glucose 6-phosphate. And then the atoms rearrange in step 2, and the enzyme helping the atoms rearrange is phosphohexase isomerase. So once the molecule is rearranged a little bit, it looks the same. See, it looks exactly the same as glucose 6-phosphate, but that's because the unwritten atoms in this molecule, because see, we got stuff coming off of C, like hydrogens and oxygens and stuff. And so the stuff that's not drawn on here, just for simplicity's sake, we don't draw the other stuff, but the stuff that's not drawn on here is what rearranges, which is why you can't tell the difference in the pictures between the two. But this one, after it's rearranged, is not called glucose 6-phosphate anymore. It changes into fructose 6-phosphate. So see, it's almost the exact same name, but that glucose changed into fructose, which is an isomer of sugar. So in step 3, we start with fructose 6-phosphate, but we need to add another phosphate group now, so it doesn't slip back in its conformation back into glucose 6-phosphate. So we're adding another phosphate to the other end of it, and remember, this is carbon 1, carbon 2, 3, 4, 5, 6. We already had this phosphate attached to carbon number 6, which is why it was called fructose 6-phosphate. But now we're going to also have a phosphate attached to carbon number 1 as well. So 2 phosphates, 1 on each end. So that's going to be named fructose 1, 6 bisphosphate. The bis just is another way of saying that there's 2 of them, but they're not next to each other on the carbon chain. And the 1, 6 means those 2 phosphates, one is attached at the 1 carbon and one's attached at the 6 carbon. And don't freak out if that's kind of foggy or hazy for you or kind of hard to understand, because you get more into this when you take something like organic chemistry. So don't sweat this too much if this is for your like Bio 101 or Microbiome 101. Chances are you're not going to need to memorize all of these, or maybe there's like one your teacher in particular wants you to know. So the enzyme responsible for this is phosphofructokinase 1. And it's kinase 1 because it's adding a phosphate to the first carbon. And that's just abbreviated PFK1. So here in the box, PFK1 is the enzyme responsible for adding another phosphate group to this fructose 6-phosphate, but this time to the first carbon, which is why it's PFK1. So now we start step 4 with fructose 1, 6 bisphosphate. But I divided up step 4 into 2 separate kind of sub-steps. So this is step 4A. This is the first half of step 4, which is this molecule splits right down the middle. And so you end up with mirror images of each other. You end up with a 3-carbon chain, 1, 2, 3, where the phosphate group is attached to the 1, and this is called DHAP. And you end up with another 3-carbon chain, and this is 1, 2, 3, keep the numbers the same, and this phosphate group is attached to carbon number 3. So they're mirror images of each other. DHAP has the phosphate group attached to the carbon number 1 on the left. On the right, GA3P is the carbon attached to the 3, the third carbon. The phosphate is attached to the third carbon on the right. And the enzyme responsible for this is aldolase. Here is the second half of step 4. So this was the new molecule on the left. See, 1, 2, 3. The phosphate group is attached to the 1, carbon number 1. So that's the DHAP molecule. So this needs to change into that mirror image molecule, that GA3P. So it rearranges so it can just convert to the GA3P. So now we have 2 molecules of GA3P, or sometimes it's just called G3P. It's the same thing. And the enzyme responsible for this is triose phosphate isomerase. Because again, this changed into an isomer of itself, the mirror image of itself. So now we have 2 of these, 2 molecules of carbon, carbon, carbon phosphate. And so we need to add phosphate to the other end so it won't slip back into its previous conformation. So the enzyme responsible for that is GA3P dehydrogenase. And this time, instead of ATP and ADP, the energy transporter that is going to come in is the NAD+. And it's going to swoop in and pick up a hydrate, which is a negative hydrogen, and it's going to turn into NADH, which is what enables this extra phosphate to come in and attach to the other end of the one carbon, 1, 2, 3. So that way we have a phosphate added to both ends, again, bookending the carbon chain. Now, this molecule with the phosphates, the two phosphates, one on each end of this 3-carbon chain, since we have carbon 1, carbon 2, and carbon number 3, we have a phosphate attached to carbon number 1, and we have a phosphate attached to carbon number 3. So this is going to be called 1,3 bisphosphoglycerate. And the phosphoglycerate kinase is the enzyme responsible for ADP coming in to pick up a phosphate, and we're going to lose this left phosphate off of the 1, leaving only the carbon chain with the phosphate group on the carbon number 3. So now we're losing phosphates, we're taking them off. The first half of glycolysis, we're adding phosphates on. The second half of glycolysis, we're taking them off. So now we have this, we had 1,3 phosphoglycerate. So now we just have 3 phosphoglycerate because we took off the phosphate that was on the first carbon. We took that off in the last step. So now we just have a phosphate attached to number 3. So now it rearranges, and now the phosphate jumps from carbon number 3 to the middle carbon, carbon number 2. So now we're going to call that 2-phosphoglycerate. And the enzyme for this is phosphoglycerate mutase. So now we have 2-phosphoglycerate because the phosphate is attached to carbon number 2 on this chain, and it needs to rearrange the phosphate on the molecule so there's an oxygen here in between carbon number 2 and the phosphate group. So this is going to be called, we call it PEP for short, phosphoenol pyruvate, after it's rearranged and we have this oxygen separating the phosphate group from the carbon number 2. So the enol means it's been rearranged, but it's still made up of the same stuff, just rearranged a little bit, the molecule is. So now in step 9, we have this, there's oxygen here, and then the phosphate group attached to that. Now we just get rid of this phosphate, and to get rid of it, we use the enzyme pyruvate kinase, and it gets rid of the phosphate, that little arm sticking out, and so now we just have the 3-carbon chain again. And this 3-carbon chain, now that all the rearranging is done through ADP, picking up the phosphate and turning it into ATP, now this is our pyruvate. And remember, all of these happen in 2s because remember we divided that up. We divided up the molecule a couple of steps ago into 2 molecules. See, in step 4, we split this molecule in half, and then we ended up with these little 3-carbon molecules instead of a 6-carbon. So now we have 2 of everything. So everything after this is going to be 2 molecules this is happening to. And so here's the chart again, and let's just do a quick review. So now that we've gone through it, you might be able to understand it better. So we start off with glucose, and it enters the cell, and we have to attach a phosphate group to it so it stays in the cell, doesn't slip back. And we do that because the ATP drops off its kid at school, right? And so that's the phosphate group that is dropped off here. And it changes names, and then it rearranges itself, and it changes names again into fructose 6-phosphate. And then we use ATP again to drop off another kid at school and to attach another phosphate to the beginning of the carbon chain. Then it divides in half, so you get mirror images of each other. This mirror image on the left mutates, or not mutates, but transforms into, it rearranges so it's exactly the same as this molecule on the right. So now we have 2 of these. So everything from here on out happens twice. Now we do the extra phosphate with the NAD plus comes in, removes a negative hydrate and a negative hydrogen, and the phosphate comes in and attaches itself to the other end of that carbon chain. So now we have phosphate on either end. And then we start removing the phosphates. So we get rid of this with the ADP coming in, picking its kid up from school. And then the phosphate that's still left rearranges so it's in the middle. And then it rearranges again. So it's got like a little arm there. And then ADP comes in again to pick up its kid from school, picks up that extra phosphate. We get rid of that phosphate. So now we just have this 3-carbon chain called pyruvate. But there's 2 of these, remember. There's 2 of these carbon chains of pyruvate because we divided this big glucose molecule in half earlier. So the results of glycolysis, and this is something you are going to probably need to know, it's usually a test question, are at the end of each of these stages, the glycolysis stage, the transition stage, the Krebs cycle, and the electron transport chain, what are the net results of each of those levels, of each of those processes. So in this one, we end up generating 2 ATPs. We generate 2 ATPs because we're including both molecules after they're divided in half. So it generates 2 ATPs, it generates 2 NADHs, and that in turn generates ultimately 2 pyruvate molecules. So to recap, in these glycolysis steps, I broke it down so it's really simple. The first step of glycolysis is it enters the cell and we add a phosphate to keep it from slipping out. So to add the phosphate, we use up 1 ATP, 1 ATP used. We use up 1 ATP to add a phosphate to the glucose. And then it rearranges, so we don't need an energy carrier when it rearranges. And then we add another phosphate to the other side of it. So that's going to be another ATP used. And then we split the molecule in two, so now everything's going to happen in twos. So we add a phosphate, but this time when we add a phosphate, it's because we're adding it with NAD, which gives us NADH. But remember, now that we have the split into two, so we're doing this 2 times with the 2 different molecules, the 2 different halves of the molecules that we split up. So NADH times 2 is gained. And you can also say here, 1 ADP is gained, and 1 ADP is gained. But we normally don't count the ADPs because we only count the ATPs because the ATPs are with the energy carriers. The ADPs are depleted, they're without energy. So we don't worry about these. We just count the ATPs used and gained, just like we count the NADHs gained. So we have, so far, we have 2 ATPs used up and 2 NADHs gained. Then we need to remove a phosphate, and we're going to do that by ADP swooping in and picking up its kid from school. So ATP gained, but remember we're doing this twice now because we had divided that molecule in half. So 2 ATPs are gained. Then we need to rearrange, and then we remove the last phosphate with another ADP, and so giving us ATP. But remember there's 2 molecules here now, so we're going to take that times 2. So 2 more ATPs are gained. So in all, we have 2 ATPs here, 2 ATPs here, but then we subtract an ATP that we used up here, and we subtract an ATP that we used up here. So 2 plus 2 minus 1 gives us a net result of 2 ATPs gained. And we also gained 2 NADHs. And of course, the 2 pyruvates. So this might be a test question. How many ATPs or what is gained at the end of glycolysis? So you'll need to know the 2 ATPs, 2 NADHs, and 2 pyruvates. And that's it. That's it for glycolysis. And if you have any questions on this, I know it can be a little overwhelming when you first get into it, but it's really not bad if you go over it a couple of times, then you'll catch on more and more. But please feel free to leave a comment in the comment section if you have any questions, or refer to my website and you can email me through that.